Precisely resolve the subcellular topology and compartmentalization. Image spatially resolved protein and RNA localization and co-localization. Precise spatial recovery allows you to track single molecules in live samples and quantitate your observations. Cells and molecules don't exist in isolation. They form large, interdependent, and dynamic networks. But how can we ask precise questions in this world of vast biological complexity? Double Helix Optics allows you to harness the true potential of spatial biology with its range of spindle devices and recovery software that will add a new dimension to your research. Simply slot the suitable phase mask into the spindle module, attach it straight to your standard microscopy setup, and you are ready to take your experimental observations to the next level. Faster imaging, reduced phototoxicity, and significantly smaller data sets to analyze and store. Our technology is applicable in a wide range of life sciences research topics, from studying single molecules through cellular and all the way to systems biology. Embrace the depth and complexity of spatial imaging and unlock your next discovery with Double Helix Optics. What will you discover?
Oops, how do you get out of here? There we go. Okay, so you know a little bit about us. We're based out of Boulder, Colorado. One thing that's really important in our field is our patents, because you can do imaging in a lot of ways, and you guys can teach me about how to do imaging, I'm sure. But we've chosen a particular path, and it's really important to patent what you're doing if you think it's novel and you want to commercialize it, because that's really how people say, okay, this is something that's distinguishable. This is something that if I commercialize, it can protect. And it also says it's novel, right? And so we're really excited about our patents. And we're also excited by the fact that this technology came out of collaboration between my co-founder, Dr. Rafael Piastun at the University of Colorado Boulder, who is a specialist in computational optics and 3D imaging, and W. Morner, Bill Morner of Stanford University, who is in chemistry and won the Nobel Prize as one of the inventors of super resolution microscopy with his single molecule technique. And so when you think about it, why would a guy who did a PhD in imaging, 3D imaging, end up with a guy who did chemistry? And that's the power of collaboration. And so I'm sure you guys cross-pollinate around here, but I really, really strongly recommend it, not only in this building, but I think there's another science building here too. So, but it's amazing what great minds put together can create, right? So the other thing that's really important for us is the validation of the technology, not only because we say it's great, but because people at your level who are rigorous and are only going to publish if things make a difference say it's also good. So what's really exciting about this is if you look on our website, there's been papers written not only in the biological space, but in the metrology space, metamaterials, AI, machine learning. So you can really use this in a lot of dimensions. And that is one of the reasons we think from a commercial perspective, it has a lot of value. So here's how we see the world, that we can potentially disrupt it, or we will disrupt it, I should say. That's what an entrepreneur says, we will disrupt it in a number of areas. We started in the area of biology, in cellular biology, and now we're moving into multi-omics and next-gen sequencing, which are very large, interesting markets on the life sciences side. We more recently introduced products for the inspection, both surface measurement and what we call multifocal object inspection, meaning we do multiple planes in a single image. And then one of the really interesting, nice benefits of our 3D technology is that we can actually help with some 2D optical correction. And why is that? Because if things aren't flat, and I use a bacteria as an example, you can use anything that's kind of got rounded edges. If you're in a 2D focal plane, you kind of don't get those rounded edges because they're not flat here. But because we see through space, essentially, we capture the whole thing, whether it's flat or curved or off center or off kilter, right? So that's kind of a nice benefit of our technology. So we think about the world and what are the problems that people are trying to solve? What are the challenges? So first thing is the world is seeing smaller and smaller things, whether it's objects for inspection or it's life sciences. So you want to see the full 3D structure. And as WE said to me very early in double helix's lifetime, Leslie, even something as small as a single molecule is inherently 3D. So the world is 3D. People say, well, I can't get depth because I need resolution. Can you improve my resolution? Well, I can in some ways with some algorithms, but generally speaking, I'm not going to enhance your depth and get you better resolution, but I can make sure that you don't lose resolution. Now, is it exactly, do you lose 0? No, but you'll see later that it's a very minimal loss and it can be recovered. Focus precision. So this idea of instruments that they're kind of complicated. We all know that if you buy a microscope and you look at what the specs are, oftentimes, particularly the objective's a little off spec. It's not exactly an NA of 1.45 or 1.49 or 0.45. So we say it doesn't matter because we're in focus above and below that nominal focal plane. We take away the need for everything to work perfectly. We take away the need for the biologist to put the sample in exactly flat. So these are some things that are really nice, and we go fast. If I can take the same data by reducing the number of images you have to take by 3, 5, 10 times, what does that mean for the speed of acquisition, and what does it mean for the data sets and then that you have to analyze? It's multiplicative, right? If I reduce it by 5 times, you're going to have 5 times smaller data sets. So we do this in 2 ways. We call it deep focus. So 3D our way. So we have conventional optics where you can see that you're in focus at the nominal focal plane, but then things blur out pretty quickly. We have our extended depth phase mask. So this is what we call volumetric recovery. So you're essentially seeing an X and Y. I mean, you're seeing the whole volume in X and Y. I'm not giving you a Z coordinate, but I'm allowing you to see the full image. And that's in focus above and below. And so you can see now your green and blues in focus. And then we have our 3D phase mask, and this is where the double helix name came from. That's our original phase mask, where we actually will give you XYZ coordinate information for every point that you're looking at. Okay. Let's see. Okay. So here's our 3D PSF. So this is our double helix PSF. So the idea here is this is a standard PSF, and you can see it's coming in and out of focus. And you can see that the double helix PSF is staying in focus, right? Because that's through depth. That's what this is showing you. And you can see the rotation of the lobes. So as the lobes rotate, that's telling you where you are in the Z position. So how deep are you in that sample? And the midpoint between those 2 lobes is giving your X and Y coordinates. So the idea here is you can get this information. We can match the depth precision to what you need. I mean, so maybe you need 2 microns if you're studying bacteria, or if you're studying T cells, you might need 13 or 15 microns, right? But you don't want to redistribute the light beyond what you need to recover what you're looking for, right? So I don't want to give you one depth extension and say, oh, this is the universal truth, which is kind of what astigmatic does or cylindrical in does. It is chromatic, so they have, we fabricate these to, to certain wavelengths, and in biology, there's certain fluorophores that people use or quantum dots or whatever it is, and we know what the peak signal they're looking for, what the emission wavelength is on the laser, and we can match to that, and then you have about 100 nanometer band, plus or minus 50 on either side. Let's see. And here, I don't know if you're familiar with Kimerling lower bound, but this is how we measure precision so that we know what we're getting. And the idea is we're getting precision in X, Y, and Z. So I'm not trading off precision in X and Y to get precision in Z. I'm going to give it to you in all 3 dimensions. And I'm going to give you the depth extension. And here's a really pretty example, we think, showing you our different depth extensions, right? So this is a nuclear lamina. It is about 5 microns of depth. But you can see we used a DH PSF that had 1 micron of depth, second one that had 3 microns, and one that had 6 microns. And the idea here is you're going to get your precision, whether you're doing 1, 3, or 6. So depending on what you need, in this case, it's kind of lovely to show how consistent it is through depth with your resolution. It's a nuclear landmine in a U2OS cell. I don't know what a U2OS cell is, but the nuclear landmine is that nucleus part of the cell structure, right, where a lot of the stuff is going on that they want to see the composition.
In the speckling we see an indication of the spatial resolution of the scale, or?
Yeah, so the depth is encoded in color.
But I mean, the speckling, the dots, like in the far right, it's very clear there's lots of individual dots. Is that an indication of the spatial resolution of the sensor?
This is, yeah, so this is, you're seeing a super resolution image, so those are individual molecules inside of this nuclear lamina. So that's why you're seeing all the spots.
Okay, cool.
Okay, so now this is our extended depth where you don't need XYZ coordinates, and that, think about metrology. A lot of times you don't, if you're just looking at a defect, or even in high content screening or other applications in life sciences where you want to go really fast, you're looking for something, but you don't necessarily have time to wait and find what the XYZ coordinate is of every protein or every RNA. You just want to see if it's there. Is it present? So these are our extended depth mask. And here again, you can see the standard PSF. You can see it's a different shaped PSF here, because now it's different shapes for different returns, different results. And now we're just extending the depth, 2, 3, 5, 10 times type thing. This is not chromatic, so you can use it with any, from 400 to near IR, and I think you can even go into IR. We haven't really done that, but that's what's nice about these. You don't have to swap them out based on whatever lasers or fluorophores you're using. And here, this is, how do we get this? This is your cost. So here we're looking at peak signal versus Z. So when you focus the light, as you know, on that very narrow spot, which is clear aperture is in the blue, then you can see it has a high peak signal compared to our phase mask, which are in all the other colors below. But the thing is that we're actually staying in focus through Z, right? So we do better through Z and signal than clear aperture does. Does that make sense? And these are just different depth extension masks. And so we can recover. So it's about a 5 or 10% light loss. Doesn't really matter in a lot of applications, but you can also recover it using computational recovery. So here's an example of using this deep focus mask, this extended depth mask. Here's clear aperture in the left. So this is 5 slices, and you're using a 5 slice MIP to kind of average Z. And this is what you get with clear aperture. The image in the middle is the double helix using our technology, taking that same protein and a single image, or this is actually proteins in tissue, so it's multiple proteins. But you can see that it's pretty clear. I mean, there's no computational recovery. This is straight out of the microscope. This is exactly what you see. And there's no additional time for acquisition time. So if it's 15 milliseconds for clear aperture, it's 15 milliseconds for double helix. But then what's really nice is through computational recovery, we can even enhance resolution. So people often say, can you get us better resolution? The answer is yes, with computational recovery. People say, how long does it take? Well, we go pretty fast, right? Our acquisition times are as fast as your camera, okay? And our computational recovery, I think if NVIDIA wanted to give us their super duper computer for free, I could probably even do it faster than we do it. But the point is, we can go as fast as we want to spend the money on the computational recovery in terms of the hardware, right? It's not that the algorithm is complicated and lags and takes a lot of time. So that's what's pretty nice. We can keep getting faster and better as we put more hardware and more stuff on there. But for start, we don't invest in like the latest and greatest, because as you guys know, it's kind of like a car. As soon as you take it out of the shop, it depreciates by a lot. So might as well. Yeah.
Can you describe briefly the computational recovery that's used here? Where is the information extracted from?
So we so we know the shape of every PSF, right? What's that?
Three-dimensional.
Yeah. So we know the shape of, you know how in the prior slide I showed you the shapes. So we know what those are. So we know what we're finding. And so we just run it through our algorithm, which is kind of our, in this case, it's a form of deconvolution. And then we can recover. And the a priori knowledge of the shape of the PSF is going to be very important as we move into the world of machine learning and AI, for example. Here's an example of RNA where we were looking at the customer is in the spatial omics domain and they have a machine and it takes them 24 hours to do a run. So can we make that faster and can we get more quality data? Because that's what they're looking for. And so the gains using our technology were, again, we just put our double helix phase mass using our spindle product, attached it to their microscope, and we were able to recover 78% more spots in tissue and 94% more spots in cells. So tissue is denser than cell, so it's a little harder, but I would say a 78% improvement is pretty good. And remember, we're replacing, in this case, C steps of 500 nanometers with a single image of 3.5 to 4 microns. And this is the core of what we do. So, you know, we have this library of mass, we have different ones, different depth extensions. So we can, in our volumetric, we go 2 to 6 times, but we can go deeper. Just most of the applications that people have asked for haven't needed more than 6 times. And in the 3D, we can go 2 to 20 times. Now, what's the limiting factor? Sometimes it's the signal that you're getting out of your sample. So yes, I can go 20 times. Does that make sense? Given your sample and given your biology, no, it doesn't make sense, right? So we work with our customers to really identify what's the optimal for you based on what you're trying to accomplish with your imaging.
When you put on deeper depth, do you have to increase the NA of the system?
No,
So it's the same optical, the imaging system, the only thing changed is the mask.
that's right. Right, because they're choosing an NA based on some of their own criterion that are kind of independent of double helix, right?
What produces the two areas?
The double helix, the 3D. Yeah. So here you go, the double helix used in different cases, 2 microns, 5 microns, and 12 microns, right? So again, a T cell is different than the cilium of cell or bacteria, right? So why give you a 12 micron mask if you only need 2 microns of recovery? And why give you 2 microns of recovery if you need 12? So we are a computational optics company. We value extracting the data with our mask hardware, but then you got to interpret it. And we want to give people back information, right? We don't want to just say, here's an image, good luck, figure out what it means. And so we develop a portfolio of software tools. We design them as modules so that depending on what you're doing, so if you're doing super resolution imaging, it's one module. If you're doing tracking, it's a different module. If you're doing counting, it's yet another module. If you're doing wholesale reconstruction or let's say inspection, deconvolution type stuff, we have 2D and 3D deconvolution. So we really want to match what you're doing to your application. And then we also have people who work in sparse samples and people who work in denser samples. So you understand that one imaging thing doesn't solve all things. And so you got to sort of, you know, I wouldn't say customize it, but optimize it. And we do that through our algorithms. And then, of course, you can't stop with algorithms. You got to go into the machine learning. And today at lunch, we were discussing what's the difference between machine learning and AI. And I warned the students today that this was going to be called 3dtracks.ai because that's what's sexy. But you can think about it as a machine learning algorithm, right? We know what the PSF looks like. We know what we're looking for as we get deeper into the sample and it gets a little hazy. We kind of know what's background. And one of the side benefits of the technology is because we're going deeper, we actually reduce the background because we're in focus over more Z. And so we can actually separate out the object from the background a little bit better than if you're just doing 2D scanning. And so there's a paper coming out with one of our collaborator labs at Rice University that took all these double helices, validated them against different depths to show that the resolution was consistent with depth extension, and then also applied AI and was able to get to image 10 times denser samples faster than if you didn't use AI, if you just used our regular recovery algorithm. So we're really excited about bringing that to the market as well. And what we just want to show here is this isn't theoretical. This technology is being used in the marketplace today, whether you're looking at, from a farmer perspective, drug discovery, you want to understand the basics of particular scientific areas, or in this case, biomedical engineering. And I couldn't even tell you exactly how they're all using it, except it's exciting that they need the spatial recovery at the precision and depth that we require. And then the other thing that's really cool about what we do is we're not tied to a particular illumination approach, right? So we might have started in wide field, but you can see we're being used in cryo-chem now, which is really exciting because now you really want to, EM is great. It's very, very precise, but once you blast your sample, you blew it all up, right? And if you didn't blow it up in the right place, then you got to start over again, and it takes time and it's expensive. But with our technology, we're able to allow you to identify where is the protein of interest. What do you care about? Because we're giving you that single molecule sensitivity. And so we're able to see where we want to go in that sample to do the ablation. So to us, that's a very exciting application because it's just, again, it's incremental to good techniques that are already there. We're not saying throw out your EM. We're saying let's get you a better result using your EM. Light sheet is a wonderful thing if you care about phototoxicity, if you really want to be careful about bleaching out your sample, particularly if you're using live cell also. And the idea is we can now bring single molecule sensitivity, and most light sheet today has been sort of organoid and larger in terms of application. And then, as I mentioned, deep learning. But we don't want to stop with biology, as exciting as it is, and there's a whole future for us there. We wanted to extend the capability of our technology to larger objects. So some companies start at the top of the funnel and they get more precise. We start at the bottom of the funnel and we're getting bigger. And the nice thing is we know we can do it because we did it at the molecular level. Why can't we do it at the target level, right? Something that you can actually hold in your hand and see. So same principles, same mass, different sizes. And here's what we think is so exciting. So you've probably used Air Force targets or seen Air Force targets. So we took Air Force targets and we spaced them, and then we rotated one so they're kind of like this to each other. And when you do that and you try to take an image using clear aperture, well, you can either see the top or you can see the bottom. But you can't see both simultaneously. The whole thing looks like a blur, right? But insert our face mask and voila, you can see above and below and everything in between. So it's really, really exciting because it's that simple. We changed the size of the mask so that, from a Fourier optics perspective, we were in the pupil plane and we were covering, we're sized to the numerical aperture of the objective, and they typically use lower NA objectives in inspection, but it's not a new design. It's just a new application. So it's really exciting for us. And now this is sort of our trajectory, right? I talk a lot about the phase mask because that's the core of our patents. That's where we started. But a phase mask will only get you so far in the world of commercial life. So we had to build modules to say, here's how it works. Let me show you that it actually does what we say it does. And so we built our spindle module, which is the single channel relay system where we can slot in the mask, and we designed it with corrective optics in the front. So we could attach to any microscope, and if the objective was a little off, we could adjust ours to be just right? We weren't going to make it fixed. And then we have these masks that slot in and out. I should have brought the module with me. But right here, you can see this is magnetic, and so you can slot the mask in and out based on what you need to do. And if you don't want to use it at all, you can just we take the mask out, we put a cover on it to bypass mode, and you don't have to take it off your microscope system. And in biology, they oftentimes one color is not enough, and sequential sequential multicolor is not good. They want simultaneous multicolor. So we built our spindle squared module, which is a single camera where we split the light path, and now you're doing 2 channels. So you can either put it in single channel mode, dual channel mode, bypass mode. You can do something. You can do polarizing in one channel and imaging in another. You can do imaging in one channel, tracking in another. It's really meant to say, go explore the world and use us as the best you can. We're not going to constrain you. And then along the way, we've been building this 3D track software. We then applied, as I said, to inspection metrology. And in inspection metrology, they don't really like these modules. As small as they are, they're still too big. So we made this thing. We call it a microspindle. And essentially, we put the phase mask in this element, and we screw it to the back of the objective. And that works with lower NA objectives because they're not quite as complex as higher NA objectives. And so it's a beautiful, elegant, and simple, cost-effective solution. But we weren't satisfied with that. So now we're really, really, really excited about this, which is we're actually putting the phase mask in the objective. So now you have a 3D objective. I don't think anyone's done that in 350 years plus of microscopy. So it's really, really exciting. So this is our module. You kind of see, I just put this here so you can see the size. It's not huge. I mean, you all have optical. I saw a couple optical tables today. You know, it's not like you have to go buy a whole new optical table or a whole new system. These can, if you have a little room on your table, these can slot into the C mount or the F mount of your microscope, and we just sit in between the camera and the microscope, and you have your spindle system installed, right? And that's the 3D track software there. Here's the microspindle. You can see this is our microspindle and our in-spindle, our inspection spindle. Because again, inspection metrology, they don't want as complicated a system as what life sciences needs. So we're like, that's fine. We don't have to give you a complicated system. We'll give you a simplified system. And as you can see, this is just, here's the objective here, and we just screwed it into the back of the objective, right? And so we can swap out masks, or we just sell you a bunch of these different ones with different masks in, and you can screw them in and out, right? So the one thing I do want to say is with this thing here, we just took it up to a partner lab in Colorado, and we were able to install it in 10 minutes and align it and start to do imaging. And an hour later, we were out of the person's lab, and we had the 3D data and the images that they wanted. And to me, that's amazing, because you often think of optics as complicated and complex, and you have to swap out your workflow and all this kind of stuff. And imagine, I mean, I could walk around the world now with this. I don't even have to check bags. It's exciting. It's not a cheap Nikon objective.
When you were able to find the pupil plate, actually there was space in there. Glass.
Well, that's the thing. We did it on, we attached it right here at the edge. The next step is to crack open. That's the next step, right? And probably what we'll do is partner with objective company instead of cracking open what's already been made. Build the model with them and build it in before they encase it. Yeah. But we did it without having to crack open. I mean, think about that. That's kind of cool. Okay, so that's just to remind you of what we do, which I think I've said enough times. But here, the last question I was asked in my talk was to answer, you know, what comes next? If we've done this, what's next? So I thought about it. Clearly, I was a little confused. Anyway, here's what I said, and it kind of goes to the conversation earlier today I had, which is it's seeing as if the human eye. Maybe I'm not exactly right that we were humanized 2D, but if you'll let me have a little business grace in terms of my statement, everyone kind of wants to see how we see in the world. We don't want to see it flat, and we don't want to see it in black and white. So while we're focusing on metrology and inspection and in life sciences, you know, Apple has now coined what you would call AR/VR a while ago to spatial computing. And if Apple names something, you know, it's going to be, that's just what it's going to be in about 10 years. So that's what it is. And then, you know, autonomous driving. Well, we're not quite there yet, but what are we trying to do? We're trying to act like we're the driver and you don't need to drive, which I don't know will ever happen, but that is what people want, right? So we're not going to do that, but I think what I think is that we're going to see better than with 20/20 vision, because what we're allowing you to see are things that you can't see with human eye and in 3D. And so that's really exciting. So I think that is it. And now questions. Yes.
You may have said this and I missed, what's your wavelength? Can you go into the infrared?
So to date, we've gone like from about 400 nanometers to near IR. I don't think there's any reason I don't know that there's any reason we can't go further. It's just we haven't been asked. So. Yes.
So the video showed what looked like a cartoon animation of exploring some theoretical world. Have you done something similar with real data?
Yeah. Yeah. So I think that was back here. So what that was showing was taking you through tissue, right? Where you're looking at RNA and protein. So this is protein in tissue. This is in situ, right? So this is proteomics. And this is looking at RNA in tissue, which would be genomics, right? And then oh, well, you know, it's really hard and PowerPoint, Canva, I mean, to do everything in 3D. I think that's why we showed you some of those movies to show you the 3D. Did you see the movie with the rotation? The point here is this, yes, it's very hard to see with, I did the pretty images as opposed to the MATLAB images where I could have sort of showed you through. But this is essentially saying this is one image here on the right, and the standard imaging is about 8 slices, right? But we're showing you it. I guess I could put more movies in. I don't know if that satisfies the answer. Depends on what your role is in the company. Like my partner, Rafael, doesn't have an MBA and he has no interest in an MBA. He likes being the professor who is thinking about the physics and the science, right? My recommendation is if your background is in the technical side, find yourself a business partner who can think about the other side. And be open to learning along the way. But to start, you don't necessarily have to. It's really about the team that you form, is what I would say.
I will ask that, face mask is face only mask means it only have face. I'm curious about that, 3D PSF, how you find that? That's ePSF.
Oh, it's called engineered point spread. We're engineering the point spread function. Right, because it's the native point spread function is this, the standard PSF. What people understand, most PSFs, not maybe in this room, but in general, the PSF is just this spot, this airy PSF. Right, that's kind of the standard PSF that you find in every microscope, right? So we engineer the point spread function to give you a different response. That's why we call it ePSF.
And this is the variation of different PSF, right? You have generated.
Yeah. Right. So this is our engineered 3D PSF. The double helix, the rotation. That's right. So when you insert the mask into the pupil plane of the objective, you then, it doesn't give you this response, the standard response, it gives you this other response, right? And so at focus, the two lobes are, like this. And as I go, let's say, go into the sample this way, it rotates this way. If I'm going above the focal plane, it might rotate the other way, right? So the rotation tells me where I am in Z. Does that make sense? Yes.
Yeah, so goal of the system is to produce 3D reconstructions, or one of the goals. And I'm just wondering, when you're working on such a cellular level, especially when machine learning is involved, how do you validate that your 3D reconstruction is accurate? Right, I mean, it might look good, but there are certainly going to be inconsistencies, but maybe they're smaller than people care about. How do you measure it? Well, what's the kind of standard?
I mean, how do you set a ground truth?
Yeah, how do you compare against ground truth?
Well, I think so we can do a simulated PSF, right? So we know what it is based on the simulation and establish it as a ground truth. We can do imaging and learn kind of, recurring imaging, so we kind of know what the empirical PSF looks like for that setup before we do the AI or the machine learning. And so I think when this paper comes out, they can talk to you about ground truth, how they did it, but I think that's the two methods that we typically use. It's the simulated PSF or the empirical PSF.
So you're able to ground truth it, the optical, the imaging system, but not so much the added components from. And once you have machine learning, then you're building in additional assumptions about what the structure of the 3D world is, right?
Well, except we know the shape, so we're not really making assumptions about the structure, because we know the shape of the PSF is this. We know it's two lobes, right? Or it's that single lobe, or it's our squiggly. So we know the shape. So when we go searching, we know that we're looking for two spots, for example.
Okay, thank you.
Does that make sense?
Yeah, I guess the part that it seems like a really interesting and difficult challenge to me is that even if you know the PSF, you can validate that there's still validation left to do to account for assumptions made by the network you train for 3D reconstruction. So the PSF in that imaging comes first, but then it seems like there are additional steps required to get to the 3D structure of the object or image. And so I'm wondering, so I might be missing something, or it's just an interesting problem.
Yeah. I'm not sure I can answer it further than that.
Yeah. I appreciate that.
So I think what all the images you've shown were stained or CAD molecules, is that right? Is there a sufficient contrast enhancement to go to unstained or unlabeled cellular structures?
Label free. We've worked mostly with fluorescence, some sort of tag. Whatever they want, quantum dots. It doesn't really matter. For us, it's just a source of light, something that says I'm here, so we can find you, right? I was just gonna say, we haven't really worked in the label-free environment, but like for inspection, for example, it doesn't need to be tagged, right? Because you're getting light. Yeah.
When you use your faceplate, do you rotate? Do you ever rotate? It's static?
Can you speak up a little, G, so we can hear the question?
I was asking whether they rotate the faceplate when they use it. So it's completely static.
It's aligned. We align it,
Okay.
so it's aligned with your system.
So you have to crop your faceplate.
No, you have to align it to, you're attaching, in this case, you're attaching it to a microscope, so you want it aligned with the objective.
But mainly in a Z direction.
Yeah.
Along the optical axis. It's not like you don't care about it's rotated.
No, the rotation is reflected in where you are in the sample in Z. So are you near? If this is the focal plane and you're above the cover slip or below the cover slip, right? So that's what the rotation tells you, is where you are in the Z space, right?
In this, the two circles right now, they're in the same plane, or they're actually. have
Different planes,
Different Z.
different Z,
So you're representing different Z.
different representing different Z. That's right. Right.
Other questions from the audience? The student who have worked on this.
You don't have to use deconvolution. So in localization microscopy, you're going to use a different algorithm. So it depends on, again, it depends on what you're studying. If you're doing tracking, then there's no deconvolution, right? Because you just want to see how something moves through time and space, right? If you're doing single molecule, it's localization microscopy, so you need to localize and calibrate, right? So that you can identify those localizations and get that XYZ point for each spot. If you're doing whole cell and you're just doing that volumetric, yeah, you can, I think we showed here, this is without deconvolution. This is just native recovery. And the other one to the right is with deconvolution, our form of deconvolution. So again, it depends on how clean and clear you care. What are you looking for in the science? Right? And like I said, sometimes it's just, how many can I see? And if I can see them, that's what I care about. Sometimes it's, I want to count them. So again, and how fast do I need to go? Because like a lot of pharma work, it's very high content. They're just speeding through different assay formulations, and they want to see how many of something there is. They don't really, they don't really care about the details, right? So speed matters more than resolution, but you need enough resolution so you can identify what it is. Does that make sense? Yes.
So, you're focusing on a really small, one of the obstacles to getting really big, because you mentioned some use cases where getting much larger could make you some money.
Yeah. So we started small because of this collaboration with WE, who was studying single molecule, and then we did some proof of principle work that we realized, and I said this earlier today, kind of our sweet spot is whoever has the longest arm in this room, that 3D selfie, right? Like that's where we have an advantage over some other methodologies, because we don't like time of flight, you have to be a certain distance in order to get the information. So our benefit is you can actually be quite close to your object and get the 3D information. And sort of where we think our uniqueness lies in terms of distance from the object is from here to, let's take the long arm of Kareem Abdul-Jabbar or somebody like that, right? But that's kind of it. So from a LiDAR perspective, we won't be the ones that you put on camera sensors to sort of see where you are in space. But we did talk to a company about drones that are landing, right? And so they can sort of see the field, but then what happens if they happen to land on this rock and it kills them, right? It kills the drone, right? So we would come into focus as you're getting towards that rock and say, move, right? So that's kind of our contribution in this field. Facial recognition, gesture recognition, right? Because these objects are not far field. We're not going to be good. We've thought about security, for example, security cameras, because they're kind of out of focus. And we could probably, because we're three-dimensional, we could sort of see and things would be a bit more in focus. So those are the kinds of applications, if we went into that, what's next, would be good, we think.
So if there's no other questions, there will be a coffee break, and you still can chat or interact with the speakers. Let's thank our speaker one more time.
All right, thank you, guys.